welcome to my channel. And today continue the elongation process of replication and termination also, because the replication process is such a huge topic, so I cannot explain completely in my previous video. So I just continue it in today's video. Okay, let's start. So in my previous video I explained about the elongation and the initiation process of the replication, and now I explain about the gamma complex and the termination process. So how the gamma complex is formed? Actually, in this gamma complex is formed actually the two copies of the DNA all three, due to the presence of the two copies of the DNA all three. Okay, and this actually interact with the beta subunit clamp. Okay, this interact with the beta subunit clamp and control the attack. And this gamma complex, when the gamma complex is formed, okay, two copies of DNA all three and the beta subunit. When this gamma complex is formed during the elongation process, it helps, actually it controls the attachment and the removal of the enzyme from the template. That means it helps to interact in this process in such a way that it can control if the replication is continuing or not. Okay, replication process is continuing or not. By attaching, if the replication process is continuing, that means it attach the enzyme here, and if the replication process it when it decide the replication process is not to be continued, that means it helps the removal of the enzymes. Okay, so this is the actual role of the gamma complex. And before going to the next one, I just explain to you what is the elongation process. Elongation process is the polymerization. That means the attachment of the deoxyribonucleotide triphosphate with the 3' hydroxyl group of the RNA primer. Yeah. So now we learn about the gamma complex. Next one, after the polymerization is completed, or when the gamma complex is totally formed, then RNA primer is removed by the endonuclease activity of the DNA pol 3. DNA pol 3, 2 and 1, 3 types of DNA pol. So actually each DNA pol has the exonuclease activity. It generally removes the RNA primer. Okay, because after the formation of complete strand of the DNA, the RNA primer is not necessary. So RNA primer must be removed, okay, by the enzyme activity of DNA pol 3, and this is the exonuclease activity. And another enzyme, that is the ligase, both not only ligase, exonuclease activity of the DNA pol 3 which has, so DNA pol and the ligase both helps in the ligation process. Ligation process is actually needed mainly in the ligation of the Okazaki fragments, because the fragments are, you just check out the initiation process of the replication, then you understand what is the Okazaki fragments. So actually ligase generally needed for adding the Okazaki fragments, and here the ligase that actually acts as the adhesive. Clear? So ATP or NAD plus is also needed for the ligation process by the ligase, and during this process, or during the process continuing, PPI and NMN is released. And the last one, in case of eukaryotes, the RNA component is removed by the RNase H. Here the RNA primer generally removed by the exonuclease activity of the DNA pol. But in case of eukaryotes, this RNA primer is removed by the RNase H. And the last ribonucleotide is removed by the FEN1 or the endonucleus. Flap endonucleus. Clear? So this is the totally elongation process. Now I explain about the termination process, where the replication is totally terminated. Clear? Okay. So this is the process of termination. What is termination? Termination actually, it is invented that some sequences that is present in E. coli that is known as Terminus Region or TAR sequence. Okay, and it is about 23 BP sequence. 23 BP means base pairs. Okay, 23 base pairs is there. So TAR sequence that are invented in E. coli, this is very important because this is known as the Terminus Region. Okay, why it is Terminus Region? I will explain you that some enzyme, the enzyme is known as TAS protein. Okay, enzyme is known as TAS protein. This TAS protein recognizes particularly this TAR sequence here and binds here. Other sequence is not important for the TAS protein. Only the terminus region, where the terminus region, there is the TAR sequences, and that is recognized by the TAS protein. And this TAS protein actually binds this TAR region or TAR sequences. And what is the function of this TAS protein? It allows a replication fork to pass if the fork is moving in one direction, and we know generally replication is possible in bidirectionally. Okay, but so here what is the role of the TAS protein? It blocks to move in opposite direction. It allows the replication fork to one direction but not to the opposite direction. It blocks the way. Okay. And not only that, it also blocks the activity of the DNA helicase. And you all know that what is the role of the helicase. It helps to open the coiling. Okay. So what is the role of the TAS protein? It actually block the DNA helicase. Clearly, in that way, it stops the replication process. It occurs in such a way that the replication fork trapped and the interlocked DNA must be separated from the daughter DNA. Clear? So first, how the termination is possible? 23 BP or base pair sequence that is known as the terminus region that is invented in E. coli, and the TAS protein that actually recognizes this terminus region and binds there, and it allows the replication fork to move in one direction but blocks in the opposite direction. And why opposite direction? It needs, we learn in proofreading, because without movement of opposite direction means here some enzymes that check the proofreading process. I explained it about this, what is proofreading. So what is the role of this? It blocks the replication fork in the opposite direction. Clear? And also the role of the DNA helicase. So in that situation, the replication fork totally interlocked and the replication is stopped there, and this interlocked situation is totally cut off the daughter DNA, and now the replication is completed here. And here one important thing is that is the proofreading. What is the proofreading? During the elongation process, when the nucleotides added to form the chain, if some base pair is inappropriate to the DNA strand, in that situation, the DNA pod that has two activity, one is the endonuclease activity, another is the exonuclease activity, and it plays the exonuclease activity from 3' to 5' direction, and in that situation, it cuts off the inappropriate base pair from the DNA strand and added the appropriate base pair. Okay, and after that, after checking the DNA strand, it continues again the replication. Okay, and this is called the proofreading. So here I explain you the opposite direction. Opposite direction I explain. What is? Why opposite direction? It is needed during the proofreading process. Okay, This is important for proofreading process, because if the DNA in the DNA strand there is any inappropriate base pair is inserted, then the inappropriate base pair is totally removed by the particular that is needy or the appropriate base pair. So in that situation, if the continuing process, the replication process is continuing forwardly, but during the checking the errors, some enzyme goes to the backward direction for checking or removing the inappropriate bases. Clear? So here the replication process totally completed. This is the second part of the replication, and you just check out the first part of the replication where I explained about the initiation and the elongation process.